In this video, we will cover formation of ammonia and detoxification of ammonia. So let's start. What is the major source of ammonia? The major source of ammonia is the first step in the catabolism of amino acid where the amino group is released as ammonia. This is what the major source of ammonia. And many other sources are there like glutamic acid, deamination, amino sugar, pyrimidine, putrefaction, glutamine, asparagine. You can see in figure 14.7. And what will be its fate? Its fate will be glutamine, asparagine, and urea. Now let's see transamination. Before starting transamination, let me tell you one thing. Ammonia is very highly toxic for our body, so it needs to be removed. Otherwise it is very dangerous for our nervous system. And we will see how it get detoxified by our body in this video. So let's start transamination. What does transamination actually means? It is the exchange of alpha amino group between one alpha amino acid and another alpha keto acid. What does it forms? It forms a new alpha amino acid. As you can see in figure 14.8. Here in this figure, what is happening? Amino group is interchanged between alanine and glutamic acid, and in most of the cases we will see that amino group is accepted by alpha ketoglutarate acid, and the enzyme is aminotransferase, and the prosthetic group is pyridoxal phosphate. Now come to the biological significance of transamination. The biological significance of transaminations: first, that it is involved in non-essential amino acid synthesis. It will synthesize the non-essential amino acid. For example, pyruvate can be transaminated to synthesize alanine. And the second one is interconversion of amino acid. If amino acid number 1 is high and number 2 is low, so the amino group from number 1 may be transferred to the keto acid to give amino acid number 2 to equalize the quantity of both. This is what called equalization. There are exceptions like lysine, threonine, and proline, which are not transaminated. They follow direct degradative pathway. Now come to clinical significance of transamination. Aspartate aminotransferase and alanine aminotransferase. They both are induced by glucocorticoid. Aspartate aminotransferase is mainly increased in myocardial infarction, and alanine aminotransferase, they are increased in liver disease. Now what is transdeamination? In single line we can say transdeamination is transamination followed by oxidative deamination. In one line we can say this. As you can see in figure 14.9, oxidative deamination is taking place in liver. This is because the enzyme which is involved in this reaction is glutamate dehydrogenase, and it is only found in liver. So this reaction takes place in liver, and it needs NAD plus as coenzyme, which is activated by ADP and inhibited by GTP. It is an allosteric enzyme. This need to be remembered. And amino acids are deaminated at the rate of about 50 to 70 grams per day. Now, minor pathway of deamination. You can see in figure 14.10, that is L-amino acid oxidase will act on all the amino acid except the two. They are hydroxyamino acid and dicarboxylic amino acid. It will use a FMN as coenzyme, and the peroxide formed in this reaction is decomposed by catalase in the peroxisome. It is clearly given in figure 15.10. And one more thing, that is D-amino acid oxidase can oxidize glycine and any D-amino acid that may be formed by bacterial metabolism. It uses FAD as coenzyme. Now come to the last one, that is disposal and detoxification of ammonia. We will cover this topic under four three subdivisions, that is first line of defense, transportation of ammonia, and final disposal. As I've already told you in starting that being highly toxic, ammonia should be eliminated, should be detoxified when it is formed. So it is necessary because it is very harmful for our central nervous system. The intracellular ammonia is immediately trapped by glutamic acid to form glutamine, which is especially in the brain cell, and then this glutamine will transport it to the liver where the reaction is reversed by the enzyme glutaminase. You can see in the diagram 14.11, it is clearly that glutamic acid get converted into glutamine by enzyme glutamine synthetase and with the help of 1 ATP, and then again glutamine will convert it into glutamic acid in liver where the enzyme will be glutaminase. Now transportation of ammonia, the main we can say glutamine is the main transport form of ammonia from brain and the intestine to the liver, while alanine is the transport form from muscles. The, the glutamic acid which is forming act as a link between amino group of amino acid and ammonia. Only 2 points we have to remember in this. First about glutamine and then second about glutamic acid. Now final disposal. The ammonia from all over the bodies will reach to liver, then it will detoxified to urea by the liver cell, and then finally it will get excreted through kidney. Urea will be the end product of protein metabolism. So this need to be remembered that urea is the end product of protein metabolism. Since mammals include human beings excrete amino nitrogen mainly as urea, they are referred as ureotelic, and fishes excrete ammonia as such, so they are known as ammoniotelic, while birds and reptiles are used they excrete uric acid, so they are known as uricotelic. Now this is end of this topic. I hope you all have understood this.